Welcome to this second session on calcium dynamics. Part 2 will be about calcium-induced calcium release. My name is Jens Bangs-Jacobsen from Department of Biomedical Sciences, University of Copenhagen. First, as to the mechanisms of calcium-induced calcium release. The picture here shows how the signal molecule inositol triphosphate, abbreviated IP3, is anchored in the plasma membrane through diacylglycerol. Upon activation of a G protein-linked receptor by a signal molecule such as nor-adrenalin, activation of phospholipase C may occur, leading to cleavage of this bond between the diacylglycerol and IP3. Subsequently, IP3 diffuses into the cytoplasm and may activate a IP3 sensitive calcium channel, the IP3 receptor channel, on the surface of the SR. Activation of this channel leads to efflux of calcium from the SR lumen and out into the cytoplasm. In turn, this may lead to activation of the contractile machinery of the cell. The picture over here shows the chemical structure of IP3. The IP3 receptor channel, which is a calcium channel, has got another feature, namely that it has an activating site which can bind calcium in addition to IP3. So a small efflux from the channel of calcium may lead to further activation of channel, a positive feedback cycle, and the calcium let out in this way may diffuse to neighboring channels or clusters of channels, in turn leading to activation of those channels. Altogether, the calcium that has left the lumen of the SR contributes to the cytoplasmic pool of calcium, which is partly free and also partly buffered by specific proteins in the cytoplasm, such as calmodulin. Calcium is reuptaken into the lumen of the SR through the action of an ATP-consuming pump, the Circa pump, sarco or endoplasmatic reticulum calcium ATPase, which is responsible for keeping the concentration of calcium very high in the lumen, approximately 10,000 times higher as compared to the resting concentration here in the cytoplasm. Inside the lumen of the SR, calcium is also buffered by specific proteins such as calciquistrin. If the concentration of IP3 is low, the stimulation of these IP3 receptor channels is correspondingly low, and a small release of calcium will stay local. It will not spread to neighboring channels or neighboring clusters of channels. Such an event is known as a blip. In case IP3 is of intermediate concentration, there will be a higher degree of stimulation of the channels here, leading to a higher opening probability. So they are more sensitive to calcium. A local event can then lead to activation of the local cluster of channels, but still perhaps not activation of clusters situated further away. Activation of these channels depend on diffusion of this calcium cloud out into the surroundings. However, if IP3 is sufficiently high, neighboring clusters can be ignited, and this will lead to a wave of calcium sweeping across the cytoplasm. The kinetics of the IP3 receptor channel is described in the de Jong-Kaiser model. Each channel consists of 3 equivalent and independent subunits, and each subunit has a binding site for IP3, a binding site for calcium that causes activation, and a binding site for calcium that causes deactivation of the channel. In order for the channel to be open, the IP3 site and the activating calcium site must be occupied, whereas at the same time there must be no occupation of the inactivating calcium site. And in addition, all 3 subunits must be in this state where the first and the second site here are occupied, but there's no occupation of the third site. We denote these 3 places A, J, and K, and if the ligand is bound, whether it be IP3 or calcium, we say that the index equals 1, whereas if there is no occupation, the index is 0. So this picture shows the kinetics of the IP3 receptor calcium channel. It's a model with 8 different states, where the state can switch between these 8 different situations here, depending on whether the ligand is bound to each of the 3 places. The equation governing the transition between the different states is described by second order and first order reactions, here denoted with an A or a B. In order for the subunit to be in a state where it potentially allows opening of the channel, the IP3 site and the activating calcium site must be occupied, and this must be the case for each of the 3 subunits. Each of the edges here shows the reactions leading to and from each of the states. So, for instance, the change in the amount of subunit in this state here, where the channel can be potentially open, given that the other 2 subunits is also in this state, is described by this equation down here. So the change in this state is the reactions leading to this state. It's this reaction here, this reaction, and this reaction. The first term here describes the transition where calcium is bound to the activating site. The second term here denotes the situation where calcium is lost from the inactivating site, whereas the third term here denotes the situation where calcium is already bound to the activating site, but now there is an additional binding of IP3. All the processes leading in the opposite direction away from this state is then given by the fourth term here. Overall, the open probability of the IP3 receptor channel depends on all 3 subunits being in the open state. So, for a given IP3 and calcium concentration, the probability of being in the open state depends on the fraction of subunits that has bound IP3 and calcium at the activating site. Each subunit must then be in this state. Therefore, the probability of being in this state must be raised to the power of 3 in order for the channel to be open. Down here are some curves showing the overall picture. Calcium in the cytoplasm is here on the x-axis, the open fraction is on the y-axis, and as you can see, this is for different levels of IP3, 2 micromolar, 1.5 and 0.25. These data points here are experimental data points, whereas these curves here are curves reproduced from the model. As you can see, the open probability or the fraction of open channels increases very steeply with increasing concentrations of IP3, and the open fraction also increases for increasing concentrations of cytoplasmic calcium, but as expected, at higher levels of cytoplasmic calcium, the open probability decreases again due to occupation of the inactivating calcium site. This picture over here shows the open fraction as a function of IP3, and as you can see, there is a certain saturation of the curve when IP3 becomes sufficiently high. The de Jong-Kaiser model can be used to describe calcium oscillations in the cytoplasm using this simple expression here. So it simply says that the rate of change in cytoplasmic calcium concentration depends on efflux of calcium through the IP3 receptor channel, a leak term here, and the reuptake into the SR through the Circa pump. So the release of calcium through the IP3 receptor channel depends on this opening probability we just described with the de Jong-Kaiser model, a certain rate constant, and then the concentration difference between the SR or ER and the cytoplasm. The leak term is described simply by a rate constant and again by the concentration gradient across the SR membrane, whereas the reuptake into the SR is described by this term here that primarily depends on the concentration of calcium in the cytoplasm. Altogether, this simple model can produce spontaneous oscillations in the cytoplasmic calcium concentration. As shown in this diagram, the system will be stable for a certain range of IP3 and for a certain cytoplasmic calcium concentration, whereas there's another range here where the system is unstable and displays a limit cycle corresponding to this situation here. The de Jong-Kaiser model is rather extensive since there's a lot of different states to keep track on. It requires a simultaneous solution of very large sets of differential equations. Therefore, a simplified model has been developed, and the simplifications required for this simpler model is shown here. So first, the assumptions are that binding of calcium and IP3 to the activating site is much faster than binding of calcium to the inactivating site. The first processes can therefore be considered instantaneous relative to the third process. The dynamics of the system is then dominated by delay in the binding and release from the calcium-dependent inactivation site. This is a huge simplification relative to the de Jong-Kaiser model that has a rate constant for each individual process. The constants in this simplified model are then chosen to mimic the bell-shaped curves of the de Jong-Kaiser model, which I showed in the previous slide. And the time constant for the inactivating process, which then dominates the whole dynamics, can be chosen to reproduce specifically the wave frequency found in, for instance, vascular smooth muscle cells. This is a set of equations for the simplified model. The open probability, as for the de Jong-Kaiser model, depends on the likelihood of the activating calcium site being occupied, the likelihood of the inactivating calcium site being occupied, and the likelihood of occupation at the IP3 binding site. These two terms here are, as I mentioned before, considered to be instantaneous. So they are described by steady state kinetics. So the probability of being occupied at the activating site only depends on the calcium concentration at any time point. It's described by this Hill type equation. So you can see that when calcium increases, the likelihood of being occupied at the activating site approaches 1. Whereas when calcium decreases towards 0, the likelihood goes to 0. This term for the inactivation is described by this first order differential equation here, governed by the inactivating time constant. The first term here is the steady state kinetics of the inactivating site. That is, if we waited for a very long period with calcium concentration maintained at a certain level, then we would get this likelihood of being in the inactivated state. However, since this process is not infinitely fast, it is described by this first order equation that gives the whole system its dynamics. Altogether, we have this open probability that then enters into the equation for the change in cytoplasmic calcium concentration. So the open probability here enters into the flux term governed by the IP3 receptor channel. And as for the de Jong-Kaiser model, we have two similar terms for leak and from reuptake into the SR through the circuit channel. And this curve then shows the open probability in the simplified model at different levels of cytoplasmic calcium concentration and at different levels of cytoplasmic IP3 concentration. And here the constants were chosen so as to mimic the de Jong-Kaiser model as much as possible. But this model is much less heavy to simulate. I'd like to acknowledge professional and technical assistance from Olga Sosnovtseva from Department of Biomedical Sciences, University of Copenhagen, Denmark. Thank you.